Bismillahirrahmanirrahim. Nahmaduhu wa nusalli ala rasulihil kareem. Assalamu alaikum class. In the previous lecture, we have discussed the penicillins. In the last of that lecture, due to some technical problem, due to compression of the lecture during the uploading, some points are missed or deleted. For that reason, I am just repeating, adding those points again. That may be only the repetition, or that may be an addition. We were discussing the stereochemistry of the penicillins. In the penicillin, we have got 3 asymmetric carbon atoms, and these 3 asymmetric carbon atoms are present in naturally occurring, synthetic, and also semi-synthetic penicillins. All have got the same absolute configuration. The carbon 6, which have got the acyl amino group, has L configuration, and the carbon to which the carboxyl group is attached have got the D configuration. The acyl amino and carboxyl groups are trans to each other. 6, or substituent to the 6, is alpha orientation, and carbon having carboxyl group is called beta orientation. Then we were discussing about the mode of action or mechanism of actions. That is almost the same for penicillin and cephalosporin. We have discussed the transpeptidases, which have got the repeating units of glucosamine and muramic acid. Then we have discussed that there are certain binding points specific for penicillin and also for cephalosporin, termed as PBPs. They are from PBPs 1 to PBPs 7. PBPs 1A and 1B are transpeptidases which are involved in peptidoglycan synthesis. That is associated with cell elongation. If these transpeptidases are inhibited, they will result in spheroblast formation or rapid cell lysis take place. They may be caused by itolysins, also termed as bacterial enzymes, which are used to create nicks in the cell wall for attachment of new peptidoglycan units or for separation of daughter cells during cell division. PBP2 is a transpeptidase involved in maintaining the rod shape of bacilli. Inhibition of this results in ovoid or round form that undergo delayed lysis. PBP3 is a transpeptidase required for septum formation during cell division. Inhibition of this results in the formation of filamentous forms containing rod-shaped units that cannot be separated or it is not cleaved, but still it is not clear whether this mechanism is lethal for bacteria or not. From PBP4 to PBP6 are carboxypeptidases required for the hydrolysis of D-alanine to D-alanine terminal peptide bonds of the cross-linking peptides. Inhibition of this is not lethal for bacteria, although cleavage is required before peptide cross-linkage. Penicillin G preferably bind to PBP3, while the first generation of cephalosporins are highly binding affinity to PBP1A. There are certain specific penicillins like amdenoxylin which binds to PBP2. Then we have got certain antibiotics which may be termed as novel beta-lactam antibiotics, clavulanic acid we have already discussed, which have got the weak antibiotic activity but is a powerful and irreversible inhibitor of most beta lactamases and such used in combination with penicillins such as amoxicillin. The product will be augmentin. This reduce the amount of amoxicillin and hence reduce the toxicity, but spectrum of activity is increased. It is mainly used to reduce the gastric problems. Thiamycin is another example. This was discovered in 76 and is a potent broad spectrum, and it is used against Pseudomonas aeruginosa. This compound is useful when we are talking about the penicillins which have got certain different structures. Thiamycin have not got the sulfur in the fused ring system. Instead of that, it has got the sulfur outside the ring, or it may be termed as exocyclic sulfur atom. It also termed as carbapenems, NEMS, because it has got a double bond between the 2 and 3 positions, having the double bond termed as penems. In the same way, When we are going to study the cephalosporin, we have got the term CFAM having the fused ring structure. One is that of beta lactam ring, and the other one that is thiazolidine ring. That is, sorry, one is four-membered beta lactam ring, another one is six-membered dihydrothiazine ring. But in CFAM, having PHEM, we have got a double bond between 3 and 4th positions. Now we are specifically talking about the cephalosporins. Cephalosporins are the second major group of beta-lactam antibiotics. They were isolated in 1948 from the soil of Japan, from the sewage water. In 1961, a structure was established almost have got the same structure as that of penicillin, except having a six-membered thiazolidine ring, again, sorry, dihydrothiazine ring, beta-lactam ring, and a dihydrothiazine ring. Essential structural requirements for cephalosporins are almost same as that of penicillin. The beta-lactam ring is essential. A free carboxylic group at fourth position is essential. The bicyclic ring is essential. The stereochemistry of the side group is essential. The presence of carboxylic group at fourth position is also essential but can be converted in a group which after metabolism can generate the free carboxylic group such as can be converted into esters. In this way we can get the prodrug. If we consider the cephalosporin, SAR, we can say that there are only few positions, as in the case of penicillins. Those few positions are at 7th carbon and at 3rd position, maybe on 4th position, 4th carbon, which have got the carboxylic group, or substitution at 7-acyl amino group or carbon. Acylation of amino group increases the potentially activity against gram-positive organisms, but the activity is less for gram-negative organisms. Substitution on the aromatic ring, such as phenyl group, increases the gram positive activity, or we can say that increase in lipophilicity will increases the gram positive activity. Different flying rings such as thiophene, thiazole, pyridine, furan, amino, tetrazole can be substituted in the side chain that will result in the increase in the spectrum of activity and may also result in changes in the pharmacokinetic characters. Thiophene substitution results in a compound that is cephalothin. In the same way, furan result in kefiroxime. Substitution of tetrazole result in kefazoline and so on. Substitution on third carbon, which have got the OAC, that is oxyacid methyl group, will also affect the pharmacokinetics. The modification at this carbon 3 will reduce the degradation of cephalosporins. The benzoyl ester improves the activity against gram positive but decreases against gram negative. Pyridine imidazole replacement increases the activity against P. originosa. If replaced by azide group, decreases the gram-negative activity. Substitution of aromatic thiazoles increases the activity against gram-negative organisms, with also a better pharmacokinetic activity. Substitution of CH3 Cl will increase the oral activity. For example, when we are talking about the pharmacokinetics, if the group which is going to be metabolized is blocked, the activity will be prolonged or increase in duration of action is observed. For example, we have got a compound cephalosporin in which pyridinium group is substituted. Cephalosporin is stable to metabolism, soluble in water, low protein binding results in a good level of free drug in circulation, active against gram positive, active against gram negative, have got some kidney toxicity but at high doses, poorly absorbed, can be used parenterally. Another compound, cephalaxin, no substitution at C3, hence can be taken orally. This compound is also available in a parenteral preparation. Most of the cephalosporins are poorly absorbed. Oral absorption related to an alpha amino group at 7 position or unchanged 7 position. The substitution at C7 by a methoxy group increases the resistance to hydrolysis by beta lactamases. Example is cefoxitin. Oxidation of ring sulfur to sulfur oxide or sulfone reduces the activity, while the replacement of sulfur with oxygen will retain the activity. Compound will be oxazepam, available as latamoxif, in which activity is increased. Acylating power is also increased. Replacement of sulfur with methylene is another example, resulted in a compound loracarbef. It has got chemical stability and a longer half-life. Carbonyl group of fourth position converted to ester result in prodrugs. Increased bioavailability of sporins can be used orally. Cefuroxime axityl, cefuroxime proxetyl are the examples of prodrugs. The linkage between C3 and C4 is essential for antibacterial activity. Isomerization of double bond between 2 and 3 greatly reduces the activity. Greatly reduces the activity. If we are going to talk about the individual compound or substitution at 7-acyl group, as we have talked earlier, we can substitute thiophene. Cephalothin is one of the example in which we have got a modification at 7-acyl amino group. It is less active than penicillin G against gram-positive organisms, more active against gram-negative as compared to penicillin G, resistant to penicillinase, not active against Pseudomonas aeruginosa, poor, poor absorption, can be taken parenterally. It can be deacylate to give a free 3 hydroxyl group. Also have got less allergic reactions. Substitution of methoxy group at carbon 7 will result in cefamycin. Cefoxitin is the example in which methoxy group is substituted at this carbon or at seventh position, while it has got the thiophene in the side chain. This compound is stable to beta-lactamases, broad spectrum, but the site of injection is painful. Local anesthetics is generally used with this compound. We can also here indicate few compounds which are orally active, such as cephalexin, cephadine, cephadroxyl, cefaclor, cefuroxime oxytyl, cefixime. For parenteral use, we have got cephalothin, cefeprin, cefazoline or efazoline, cefuroxime. Parenteral cefamycin have got cefoxitin, cefotitan, cefmetazole, ceparidine, cephalosporin are orally and parenterally active. Cephalothin and cefoxitin are the most resistant to beta-lactamases. Cephaloridine and cephalozine the least resistant. The substitution of oxy-immunogroup will increase the resistance against beta-lactamases. The oxy-immunogroup or alkoxy-immunogroup NOCH3 is present in cefuroxime, cefotaxime, and also in ceftriaxone, CEFTIZOXIME. This compound have a polar function, have got methyl propionic acid